Let me start with this simple slide. Of course, it's fantastic if you have an idea for a new therapeutic modality, and then through a complex R&D process, eventually you succeed, and you have a product on the market for the benefit of the patient. For instance, here a small molecule, Xeloda, in cancer therapy, or here a biomacromolecule, which is pegylated interferon for hepatitis C treatment. So macromolecule, biomolecule, and here small molecule. Now, I'm sometimes confronted with a layman's perspective. The layman's perspective is as follows. You find somewhere from nature or whatever source a potent bioactive compound, here this powder, and pharmaceutical companies then gets it, does some testing in vivo to confirm the bioactivity and the potency, and then it formulates, and then there's the big money. Now, of course, that is not reality. As a matter of fact, the R&D process, pharmaceutical R&D process, is highly complex, multidisciplinary, a frontier science that has to be performed, and very high technology. Just a few icons here. It is high cost, long-term, and high risk. And of course, there is more failure typically than success. So, although here are two successful developments, many projects, many ideas will never come to fruition. They will stop at a later phase, or they stop already at an early phase, very early, and so on. Now, all this is, of course, pharmaceutical R&D, in the hope eventually to have a new therapeutic modality for the benefit of the patient. And when you look at it, you have, of course, the early research phase, then you have the clinical phases, 1, 2, 3, and you have an intermediate phase, typically 1 or 2 years, for the preparation to go into human. And there is a cost to the whole thing. In the research, typically compared to the development costs, these are moderate costs. But you know, if you accumulate everything, typically, and this is a very conservative value, you have $1 billion per successful drug which you can put on the market. Now, we are not subsidized by federal money, by whatever federal agency. We do not get money from the NIH. We do not get money from the Swiss National Science Foundation. We have to finance ourselves. So the success on the market, the money here is needed to reinvest into this one to make sure that our R&D efforts are sustainable for the future. Because if they were not sustainable, then of course there will be no longer any drugs coming out of this process. Now, this is often seen sort of sequential development, research and development. Here is a sort of a new idea. Then the point of intervention, the target, has to be assessed. Then there is some way of coming to a chemical compound, a lead identification. It has to be optimized with respect to its properties, particularly the pharmacological and potency properties. Then, if you are successful, these are all sorts of intermediate milestones. If you are successful, you have to do all the tests to be sure that you have no big surprises when you go into human. Then the first phase for safety, the second phase is often called the proof of concept or efficacy here, and then comes the large tests on a large population to make sure that the drug is really doing what you want and is a benefit for the patients. Then come regulatory affairs, and eventually you are, if everything goes well, on the market. Now you see a 1 here, and there have been studies, statistical studies on the major pharmaceutical companies taken together, and for each of these transitions from one phase to the next, there is a certain probability of failure, or we say attrition, of the projects. And you can calculate back, if you want to have one successful new molecule or drug on the market for whatever treatment, you need, on average, statistically, something like 14, 15 compounds that will eventually go into the phase 1. And then, due to all sorts of findings, some will drop out, the further will drop out, and eventually you have a sort of a 10-to-1 ratio from this phase here, from entry or the preparation phase, or then entry into human, to finally the market. If you want to achieve this goal here, you should start with something like 60 projects. These are not simply whatever kind of projects. These are really projects where you believe in and you have put all your heart blood into this project. Typically, our scientists have many more ideas, and this is a sort of a good guess. From some 10 ideas which a scientist may constantly produce, one project typically will make it and convince senior management and other peers that this project might fly. And that is one of those 57 or 60 projects here, which will start. And then you will discover new things when you work on it, and only 36 will survive here, and optimization. Eventually, candidates that will be treated for eventual clinical tests, you see it's 1/4 of this one. So you have a dramatic decline, attrition of the projects. But you have to start with quite many, eventually to be successful with one. Another interesting number is, typically we count on 100 to 300 scientists and technicians and so on, people, experts in the field, in a given therapeutic area, to produce on average one clinical candidate here per year. And of course, if you create one, you can be sure you will not have a full one here on the market. It's a 0.07 drug on the market. So you see you have quite a big effort and many people involved to eventually do this. There's also a timeline, and this is very aggressive, typically 3 years in this early research until clinical candidate identification, 1 year only for the preparation for the clinical studies, and 7 years for all phase 1, 2, 3, if everything goes right, until the regulatory affairs to come to the market. This is very aggressive. This is about something like 11 years. Not 1, not 2, 11. And of course there's a lead time here for all these ideas, so the lead time of 2 years before you actually start a given project. Now, this is this attrition rate, which goes down heavily. Now, today we are faced with very much more complex diseases than some decades ago. You know, we are working on schizophrenia, we're working on Alzheimer's, we're working on metabolic diseases, we're working on arthritis and so on. These are very complex diseases and difficult to treat. We have challenging requirements here. For instance, you want to have, of course, very efficacious drugs, you want to have safe drugs, you want to have drugs that are maybe personalized so that individuals that react differently are properly treated. You have higher expectations here. Today you want to have immediate action. You don't want to wait for 3 weeks or 2 weeks in antidepressives, for instance. And there are many expectations here, for instance, also once a day or once a week dosing, rather than several times a day, you know, injections, orally active, so bioavailability oral, and another expectation from society, and of course no side effects. And then higher hurdles. The more we know about human pharmacology, the more we have to do additional tests to make sure that we do not fall into any one of those newly discovered problems that may be there. There will be more and more drugs on the market, and therefore we have to make sure that our new drug will not interfere with any of the other drugs. And of course, all these hurdles make the whole process costly and more failure prone. So typically that yellow curve, due to all these pressures which are inherent in the system, push it down, and you have more attrition eventually down. And it needs no fantasy to imagine that there is no guarantee to survive. And you have seen over the last decade or so, many pharmaceutical companies have vanished, are no longer among us, because they failed to produce, to be sufficiently successful. So what can we do? We need best people, we need frontier science, we need high technology to cope with all these things which will be always here and will continue to increase pressures. to push this curve back. And I maintain against, for instance, financial analysts that sometimes ask, why with all these technologies and the whole frontier science, why are you not more productive? But I maintain that because more complex diseases, challenging requires higher requirements, higher expectations, higher hurdles, just to maintain productivity is a very big success. But there is no guarantee that we survive, and it's a continuous challenge and struggle. Of course, we would like to actually have early attrition, weed out all the projects that may fail eventually for whatever reasons, but then at this point, when we come to the clinical candidate and prepare for clinical trials and go into clinical trials, we would like to have all things in place that with all chances, these compounds will make it and will be successful on the market. So we would like to have the very expensive attrition here in this clinical development to reduce it and to have a more horizontal line. The question is how to do that, and we have no direct answer to that one, but it is a continuous struggle to try to improve the yellow curve to a sort of cyan curve here. So, I will now concentrate largely on the discovery part, this one, not on the clinical development, and want to show you how this is nowadays actually done, and I want to show you the complexity also, and also where our chemical compounds actually come from, how they are developed. So here is this white bar from the target identification, the point of intervention, the biology part here, then the chemistry part here in the middle, and then the pharmaceutical sciences, safety and toxicology part, and here